first order of business, neurogenesis. I pressed the wrong button. Okay, so for anyone who's not into the adult neurogenesis field, um, I'll let you talk about snRNA-seq because that's more your forte. So snRNA-seq analysis of the adult human dentate gyrus has been done properly and come up empty for any evidence of ongoing neurogenesis. So what this means, in broad terms, is we've always, well, there's been very strong evidence for years that when you're older, you still create new neurons. And glia have been implicated in this, astrocytes converting, like scaffolding cells in your brain, converting into new neurons. It's very difficult to test this, of course. So there have been a variety of methods. One of my favorite is from actually a lab at the Karolinska Institute where we both used to study and work, which used carbon dating of neurons. Because there was this big spike in radioactive carbon in the atmosphere during the nuclear bomb tests. And you can tell, okay, so which neurons were created during those tests and which were created afterwards. And it suggests that there is about, I don't know, something 10% turnover per year, up to. But there is considerable adult human neurogenesis. And now this study, which very, very effectively, but it demonstrated no adult human neurogenesis. So maybe all the neurons you have, that's all you've got, folks, and you won't get any more.
But you kind of caught me by surprise with this one because I don't know exactly what snRNA-seq is either. So it is RNA sequencing. So it's sequencing of the RNA. So this is like, if you take it for a single cell, it's what the cell is, like what's of the DNA is then, what is it? Translated
Transcribed?
into RNA, right? Transcribed?
I have no idea. I'm no molecular biologist.
Yeah, I'm a bad molecular biologist. I learned a bit. So I know that there's a difference between translating and transcribing. So when you go from DNA to RNA, it's one. And when you go from RNA to proteins, it's the other. But the DNA has all information to make the cell, and the RNA is the active genes, basically. So the information that is currently used by the cell. So this is what they're looking at. Let's start there. This is what they're sequencing. That is single nucleus. So they only took it from the nucleus.
Which is like the core of the cell.
Yeah, it's the nucleus, the core. Yeah. So, but this is where the DNA resides. So when you want to make proteins, you do this outside of the nucleus.
It's like woodworking. You don't do it inside the house. Your wife gets mad at you.
So now I'm thinking what this exactly means. So it's the RNA that's not transported out of the nucleus yet, probably. I wonder why they use that specifically. It's also, it might be easy to get to this RNA and to reliably sequence it.
So one thing is, this study was done in human post-mortem tissue. So these have been, patients that died of cardiovascular issues mostly. Then their brain tissue has been analyzed using snRNA-seq, and they found somehow that apparently all the neurons in their brains were old, have not been newly generated. I don't know how they checked that, but 140,000 cells, that's a pretty big data set. I gather it's quite expensive to do snRNA-seq. Let me link. So this is a bit of an older news, by the way. We haven't done a proper brains matter in a few months. So the study is called, stupid banner. I'll link it in the chat. So the study is called Transcriptomic Taxonomy and Neurogenic Trajectories of Adult Human Macaque and Pig Hippocampal and Entorhinal Cells. So this is the hippocampus and the entorhinal cortex, which is like right next to the hippocampus, usually involved in memory and spatial navigation.
Yes.
And there's no new neurons there. So if you stop being able to find your way, it's because your hippocampus does not create new neurons, maybe after all. I should emphasize, for the past 10 years, we thought there was a lot of adult neurogenesis here. This is a big finding.
So this is also just to speak against the study then, where they find that London taxi drivers have like an enlarged hippocampus or entorhinal cortex, like some area in that part of the brain, they found it to be enlarged, right? When they did scans, just an anatomical scans of brains and taxi drivers. They had this larger. So this argues for neurogenesis, like there will be more matter in this part if you use it more because you're navigating all the time, right? So
Sounds logical.
yeah. So people have argued, indeed, for a lot of plasticity, and also I think neurogenesis here. Yeah, so it's very interesting in that regard.
Yeah, I can't wait to see the next episode of scientists
Yeah.
fighting it out over whether neurogenesis is a real thing.